Assalamu alaikum and welcome everyone to our molecular biology ASCP preparation series. The MCQs we'll discuss today and in the upcoming season have been carefully collected from several years of past exams to give you the best preparation possible. I'm Dr. Laya Abdelati and I'm really excited to share my knowledge and experiences with all of you through this series. So let's dive in together. In our previous lectures, we completed the multiple choice questions related to the nucleic acid chemistry and basic molecular theory. Today, we'll move to cover the third and fourth parts of biochemical reagents, reverse transcriptase and DNA ligase. Okay, now let's focus on the questions related to the reverse transcriptase and review molecular biology ASCP questions from the previous exams. All right, the first question we have today, which virus famously uses reverse transcriptase to replicate its genome? 15 seconds. The correct answer is HIV. I mean human immunodeficiency viruses. Actually, these viruses uses reverse transcriptase to convert its RNA genome into DNA, which is then integrated into the host cell genome. Right? Let's move on. Okay, let's see the next question. Which enzyme is essential for synthesizing cDNA from messenger RNA in RT-PCR? The correct answer is reverse transcriptase. Reverse transcriptase enzyme enabling amplification of RNA sequences via PCR. All right, let's move on. All right, the next question is reverse transcriptase sensitizes DNA in which direction? 3 to 5 or 5 to 3 or both direction? All right, the correct answer is 5 prime to 3 prime direction. Actually, like all DNA polymerase, reverse transcriptase adds nucleotides to the 3 prime ends of the growing DNA strand, sensitizing in 5 prime to 3 prime direction. All right, let's move on. All right, let's see the question here. What is the common use of reverse transcriptase in research? Actually, researchers commonly use reverse transcriptase to generate cDNA from messenger RNA, allowing for the analysis of gene expression. All right, so the correct answer here is generation of complementary DNA or cDNA. All right. Okay, which of the following best describes reverse transcriptase inhibitors role in HIV treatment? The correct answer is blocks the synthesis of viral RNA. Actually, reverse transcriptase inhibitors prevent the conversion of viral RNA into DNA, thus inhibiting the replication cycle of HIV. All right. The next question. Which molecule serve as the template for reverse transcriptase? DNA, RNA, protein, or lipid? The correct answer is RNA. All right, let's move on. All right, the next question. What is the significance of reverse transcriptase in the life cycle of retroviruses? The correct answer is convert RNA into DNA. Actually, reverse transcriptase is the crucial for retroviruses as it converts their RNA genome into DNA, all right, allowing integration into the host DNA for replication. Okay, next question. Which component is essential for reverse transcriptase activity? The correct answer is magnesium ions. Reverse transcriptase require magnesium ions as the cofactor to catalyze cDNA synthesis, similar to many polymerases. All right, let's move on. All right, next question here. Which class of drugs target reverse transcriptase to combat viral infection? Reverse transcriptase inhibitors are designed to inhibit the activity of reverse transcriptase, preventing the replication of retroviruses like HIV. All right, let's move on. Next question. Which of the following is the RNA-dependent DNA synthetase? Which one of these enzymes? The correct answer is reverse transcriptase. Reverse transcriptase is an RNA-dependent DNA polymerase. It synthesizes DNA using RNA template, a key enzyme used by retroviruses like HIV to replicate their genetic material. All right, let's move on. The high mutation rate of HIV is primarily attributed to. The correct answer is lack of proofreading ability in reverse transcriptase. As we know, reverse transcriptase lacks 3' to 5' exonuclease activity, leading to uncorrected error during DNA synthesis and high genetic variability in HIV. All right. Next question. Telomerase is considered specialized reverse transcriptase because it used RNA component to extend telomerase. Actually, telomerase contain an RNA template that guides the addition of repetitive DNA sequence to the chromosome, hence mimicking reverse transcriptase activity. All right? All right. Which component is not required for reverse transcriptase to synthesize cDNA? The correct answer is RNA primer. Actually, reverse transcriptase typically use a DNA primer like oligo-DT to initiate cDNA synthesis, all right? Where RNA primers are used in the DNA replication, not reverse transcription process, all right? All right. RT-qPCR is used clinically too. Which one of them? The correct answer, quantify viral RNA load in HIV patient. Actually, RT-qPCR combines reverse transcriptase with quantitative PCR to measure RNA levels such as HIV viral load for monitoring infection. All right, let's move on. Please don't forget to subscribe to our channel to receive all the latest lecture. All right. So I also encourage everyone taking the test to share your questions with us so that everyone can benefit. All right. Let's continue. Welcome to the second part of our lectures today, DNA ligase. Let's see the first question we have. All right, the first question is, during which process is the DNA ligase most commonly used? Transcription, translation, DNA replication, or RNA processing? 15 seconds. For sure, in DNA replication, DNA ligase plays a crucial role in DNA replication by joining Okazaki fragment. Don't forget, Okazaki fragments on the lagging strands, ensuring the continuity of the DNA molecules. All right, let's move on. Next question. Which molecule is required for the catalytic activity of DNA ligase? The correct answer is ATP. All right. DNA ligase requires ATP as the energy source to catalyze the formation of phosphodiester-bond between DNA fragments. All right. This providing the energy necessary for the ligation reaction. All right. So the correct answer here is ATP. Let's move on. All right. The next question. What is the main application of DNA ligase in genetic engineering? The correct answer is gene cloning. DNA ligase is essential in gene cloning as it helps joining DNA fragments such as the insert and vectors to create recombinant DNA molecule for further study or use as we see in the image here. All right, let's move on. All right, next question. Which ligase is commonly used in molecular cloning due to its ability to join plant ends? The correct answer is T4 DNA ligase. All right, let's move on. All right, in recombinant DNA technology, DNA ligase is used to. The correct answer is join vector and insert DNA, as I said before. Ligase connects compatible ends of vector and insert DNA, creating recombinant plasmid, as we see in the image. All right. Okay, next question. Which type of bond does DNA ligase form between adjacent nucleotides? The correct answer is covalent bond. Covalent bond. All right. DNA ligase forms covalent phosphodiester bonds between adjacent nucleotides, ensuring the integrity and continuity of DNA strand. All right. Next question. In which phase of the cell cycle does DNA ligase activity peak? The correct answer is S phase. Actually, DNA ligase activity peaks during the S phase of the cell cycles when DNA replication occurs, requiring the joining of Okazaki fragments on the lagging strands. All right? The next question. Which of the following describes the role of DNA ligase in DNA repair? The correct answer is seal next and joining repair DNA strands. All right? Let's move on. Let's see the next question. What is the outcome of a deficiency in DNA ligase activity in cells? Alright, the correct answer is impaired DNA replication. Actually, a deficiency in DNA ligase activity results in impaired DNA replication, and the enzyme is the crucial for joining DNA fragments, particularly the Okazaki fragments on the lagging strands. So, this leading to incomplete DNA synthesis, right? The next question, which DNA repair mechanism primarily relies on DNA ligase? The correct answer is all of the above, as the ligase is essential in sealing next during the base excision repair, nucleotide excision repair, and mismatch repair, making it a universal component of DNA repair. Alright, let's move on. Alright, the next question. Which cofactor is required by E. coli DNA ligase but not T4 DNA ligase? The correct answer is NAT. E. coli ligase use NATs as the cofactor, while T4 DNA ligase require ATP. Okay, let's move on. Alright, the next question. In mammalian cells, which DNA ligase is primarily responsible for sealing single-strand breaks during base excision repair? The correct answer is DNA ligase 1. DNA ligase 1 seal next after DNA polymerase fill gaps during the base excision repair. Alright? Where ligase 4 involved in double strands break repair. Alright? So the correct answer here is DNA ligase 1. Let's move on. Okay, the last question we have today. DNA ligase is the critical during the G2 phase of the cell cycles 4. The correct answer is repairing replication errors before mitosis. Actually, during G2 phase, DNA ligase seals next from the repair processes, ensuring genomic integrity before cell divisions. Alright? That's all for today. I hope you enjoyed this lecture in Molecular Biology Academy channel. I look forward to hearing from you. What do you think of it? Please don't forget to like, comment if you have any questions, share and subscribe to our channel to receive all the latest lecture. Salam.